Alright, so I'm going to walk you through figure 2. And this is just an example of what I'm looking for in terms of a video for walking your audience through a figure. For you guys, for this week, it's figure 5 that you're going to walk us through. And I already showed you how to walk through the figure using PowerPoint slides if you want to. That was in my earlier videos where we were walking. I was helping you walk through the article before. So I have no problem, however you want to create this presentation, create this video to walk your audience through the figure information is perfectly fine with me. So you can create a 2 to 3 slide PowerPoint presentation where you basically are talking through different parts at a time with the take-home messages. You can walk the audience through directly on the article itself, just kind of pointing things out as you go. As long as the key take-home messages are there, and as long as the ability to show yourself kind of guiding the audience through the key aspects of the information, it really doesn't matter to me. So again, I've already shown the PowerPoint one. This is kind of the version where I'm just directly on the figure from the article itself, and I'll walk you through it. So basically, in figure 2, what the authors are trying to convince you of is that our induced pluripotent stem cells have started expressing key embryonic stem cell-like genes. So this gene expression is similar to embryonic stem cells and no longer similar to fibroblasts. So what we have here in A we basically have reverse transcription PCR. All right. And so if you had a slide, this might be where you want to just give a couple of sentences on reverse transcription PCR or something. But essentially, RT-PCR is where they have lysed open the cells. So they've taken their induced pluripotent stem cells, and you can see here that we have 5 different colonies. So these are 5 different examples of their induced pluripotent stem cells. And they took those colonies, they lysed them open, and they isolated the mRNA. So remember, mRNA is the transcription of the gene. So the DNA turns into mRNA. So the more mRNA you have, the more the gene is being expressed. Then they used PCR, all right, so they took that mRNA, they reverse transcribed it to what's called cDNA, and then using PCR, they amplified it. And so essentially, the thickness of the bands here, first of all, tell you if that gene was being expressed or not, because if there was no mRNA, then there would be nothing for those primers that are specific to that gene to amplify. And we see an example of that with OCT3/4 here with our human dermal fibroblasts. There's no gene expression there. However, we do see bands in our iPS cell colonies, and we see it in the embryonic stem cells. So this is our positive control for pluripotency with the embryonic stem cells. And this is our Entera2. This Entera2 is basically immortalized. So it's an immortalized embryonic stem cell line. So we can see that OCT3/4, and it is key to and important to note that this is the endogenous, this is the genomic version of OCT3/4, not the gene that we've added. But OCT3/4 is expressed by all of our induced pluripotent stem cells, expressed by embryonic stem cells, and by Entera2. And if we take a look at these, all right, pretty much all of these genes that we see here, so all of these genes on the line here, these are all genes involved in kind of maintaining pluripotency and embryonic stem cells. And we can see that they basically, there's a band for our embryonic stem cells amongst all of these. All right, so if we take a look here, we see that the embryonic stem cells, so here's our embryonic stem cells, the embryonic stem cells are expressing all of those genes, and the same way, so are our iPS cells. So our iPS cells, the bands look exactly like the ES cells, mostly like the Entera cells, although remember this is immortalized, so there are some abnormalities there. But key is that they no longer look like fibroblasts with regards to gene expression. So we have indeed taken these away from being fibroblasts, and now based on the expression of these genes, they are behaving more like embryonic stem cells. So that's good, but we also want to make sure that's true with the protein as well. So if we take a quick look here, this is the Western blot. So the Western blot basically is taking a look at protein. So here we're looking at protein levels. right? Over here we were looking, in A, we were looking at gene expression levels. And same idea. So here they've actually taken an antibody so that they can specifically look at the protein for each of these specific genes. So again, they lyse the cells, they get all the protein, they run the protein on a gel, but then they use antibodies for each of these specific genes, and if there's a band, that means that protein was there. And so once again, we can see that these proteins are there in our embryonic stem cell, and they are also there in our induced pluripotent stem cells, and they are not there in the fibroblasts. So once again, by protein level, our induced pluripotent stem cells look like fibroblasts. Now, if we move on to C, actually at this point, I'm going to jump down to D for a second. D basically is just looking at gene expression on the global scale. So here, they've taken the induced pluripotent stem cells, right? They've taken iPS cells from their lines, and they've analyzed the gene expression levels on a microarray of every gene at once. And they've also done that for fibroblasts and with embryonic stem cells for comparison. Now, if we have indeed made induced pluripotent stem cells into pluripotency, then we would expect them to no longer resemble the fibroblasts and now to resemble the embryonic stem cells instead. And so basically, if I took a single gene and it had a certain level in the iPS cells, so that level would be kind of down here, and then I would also measure that level in the fibroblasts. If they were the same, then you would kind of plot that on the coordinates. If they were different, for example, this one here, this gene had very high levels on the fibroblasts and very low levels in induced pluripotent stem cells. But if all of the genes had similar levels between the two cell types that we're comparing, you would expect them all to be kind of right down the center, because it would have the same gene expression level here as here. And we can see it with our induced pluripotent stem cells and our embryonic stem cells, most of these genes fall within standard deviation. They are very similar. Whereas we have a lot of outliers, they are not similar with the fibroblasts. So once again, just showing that our induced pluripotent stem cells based on gene expression act a lot like the embryonic stem cells and not like the fibroblasts. And the last thing that this figure is trying to show is the difference between the endogenous stem cell factors and the exogenous ones that we added. So remember, in order to make these, the authors added 4 transgenes: OCT3/4, SOX2, KLF, and CMYC. And in doing so, that basically de-differentiated, deprogrammed the fibroblasts and made them embryonic stem cell-like. However, we want to, even though we do want the endogenous, the genomic version of these genes to turn on, we don't want the transgene to be on forever, because if that was the case, it would prevent us from being able to re-differentiate these cells into different types. So let's say we wanted to create neurons. Well, if the transgenes of these 4 were on forevermore, they couldn't differentiate into neurons. So we can see here that the transgenes are pretty much off. They're no longer there in most of our iPS cells. This is the one outlier. That transgene's off in all of our induced pluripotent cell lines. And our control is basically showing that when we first transfected the fibroblasts, there was indeed expression, pretty high expression levels. So this shows that the genes were on, they caused dedifferentiation, and now the genes are off. So the take-home message for this figure is basically that at the gene and the protein level, our induced pluripotent stem cells behave like embryonic stem cells and no longer behave like fibroblasts. And a key aspect here is that the transgene that we added to cause this is now off, and this will allow differentiation of the induced pluripotent stem cells later on.